of the cell. We ended right here. And none of this was on the exam. We sort of ended with membrane transport. That was on the exam, remember? Facilitate diffusion, active transport. Didn't really do exocytosis, endocytosis, but there was little bits to that. We sort of stopped with organelles of the cell, and that means the organs in the cell, the little tiny organs that are found within the cell. And this is a big one because this is the control center. It means little nut, the kernel in Latin. This is the nucleus. And what do you find in the nucleus? You already know this.
Protons or electrons or
Okay, no, the nucleus of an atom, protons and neutrons. I like that. But the nucleus of a cell, good, you're bringing back chemistry. I love it. But the actual cell, what's contained in the nucleus? DNA. Okay, so the nucleic acids, we're at a higher level now. We went from atoms, now we're cells. Okay, so DNA is there, and that's what allows replication, so copying the DNA, and then transcription, turning the DNA into message, RNA, that then leaves the nucleus and comes out into the cytoplasm to turn it into protein, okay? Then it leaves through nuclear pores, and it's real small, okay, 5 microns in diameter. If we look at an actual cell, this is the surface of a nuclear envelope. All those little pores, those are the little tiny holes that allow the mRNA, the copies of the DNA genes, to exit the nucleus so that they can go out into the cytoplasm and get translated into actual protein. Here's a blow-up of one of those nuclear pores. Okay, so we'll just go through the organelle structures. The endoplasmic reticulum, this is the ER. And if it's rough and it's studded with ribosomes all over it, we call it the rough ER. That is basically the assembly line for protein production. Your little ribosomes are protein enzyme little machines. They're like the factory workers that are on the assembly line, or the rough ER, and they're what are grabbing the messenger RNA, and converting it like a typewriter into the actual protein, the polypeptide protein. What are proteins made of? Amino acids. And on that front page of the exam, you see all the pictures, the visual structures. Some of you did real good at that. Not everybody did real good at that. So if you're real visual, tend to do better. The protein, there was a protein on there. There was a couple. There was hemoglobin on there. And then there was also just a set of like 5 amino acids bonded together. So I know some people said it was a fad, or said it was it's a protein. Amino acids make up proteins, and they get bonded together through peptide bonds. That's what we make using our rough ER and our ribosomes. We also have free ribosomes that are out in the cytoplasm, and those are making proteins that are going to stay out in the cytoplasm. We have another ER called the smooth ER. Literally smooth because it doesn't have the ribosomes on it, okay? So it's tubules in this branching network, no ribosomes, no protein synthesis. Really, the smooth ER is a calcium store, and it also is involved in making fats, making lipids, fats that are going to be used, like phospholipids, that are going to be used in the cell. Where in the cell would I find a phospholipid? Many phospholipids.
membranes.
Membrane, perfect, okay? Plasma membrane of the cell. Also, the plasma membranes of a lot of these organelles. So of the vesicles, of the ER, of the Golgi, of the mitochondria, that's where we're going to find a lot of phospholipids. So here we have the rough ER, and then here we have the smooth ER. There's no ribosomes on the smooth ER, okay? But there is on the rough. Under the electron micrograph, you can see these little folds, membrane folds that make up the ER, the rough ER, and then all these little dots, almost like little, studded with little beads, those are the ribosomes. We can see this with really good transmission microscope. We can actually see the ribosomes, and that's how it was worked out. Oh, this one is rough, and this one is smooth. That's what they do. It corresponds to their functions in the cell. Now the ribosomes, if you notice, I'm using the cells almost like a factory, right? Okay, so the plasma membrane is like the barrier, it's like the fence outside. It's only letting certain things in, certain things out. It's selectively permeable. We got the nucleus, which is the command center. Okay, that's the control office. It's got all the blueprints, it's got all the instructions, it's got all the vital, critical things right in the middle. We were at the ER. The ER was what? The rough ER. What was the ER in terms of the factory analogy? Assembly line, okay? The ribosomes are what? The factory workers on the assembly line, because they're making the protein. You see that? I feel like these analogies will help it stick better. These are the assembly line workers that are actually carrying out the synthesis of newly made proteins. There's free ones in the cytoplasm, and then there's bound ones attached to the rough ER. They're made of RNA and protein, so it's this mix of rRNA, ribosomal RNA, and protein, this big complex. So they're the guys that are really responsible for plinking out all the new proteins. So they build, here's the RNA message that comes out of the nucleus. Here's a small ribosomal unit, and here's a large ribosomal subunit. They come together and they help in reading, turning the message into actual protein. And it gets built in this long chain. So they're actually zipping along, jink, 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 and reading the code of bases in the message that came from the DNA to actually make the protein. Okay, so those are the workers that are making all the protein. We have the Golgi apparatus. This is a stack of membranous sacs or envelopes, about 3 to 10 of them, and it sorts proteins that are newly made at the rough ER, and then sends those proteins to the proper places, the proper destination. Okay, so there are certain tags and sequences in these proteins, chemistry about these proteins, that the Golgi is able to, it receives them in vesicles coming from the ER, and it's able to sort them, package them, and send them where they're supposed to go. Okay, they end up going on specific tracks inside of the cell. So, a lot of times in that process, it actually alters the proteins. It adds sugars to them, adds fats to them, it makes glycolipids or lipoproteins or any of those little combinations of structures. I had an extra credit question on the exam on Friday, and it was, what are lipoproteins and what do they do? And I feel like a lot of people blanked on that, or they put nothing. They're like, oh my God, it's extra credit, thank God. I don't even remember. Lipoproteins are cholesterol, all the cholesterol we talked about. Lipoproteins are cholesterol, LDL, HDL, total cholesterol. Yep, I saw it, hardly anybody knows what these are. Yet we talked about these for like 2 weeks off and on. Well, you just didn't remember the chemical nature of those are not truly cholesterol. Cholesterol is the 4-ring structure, that's a lipid, and if we make a lot of other things from cholesterol, cholesterol's in the cell membrane. True cholesterol is different than the lipoproteins. The lipoproteins are the packages of protein lipids, they're in those big balls, and they carry the fats and cholesterol around the body, right? So there actually is more than I've taught you. There's VLDL, there's IDL, there's LDL, there's HDL. The 2 important ones, physiologically speaking, are LDL and HDL. Those are all lipoproteins. They got packaged together. They're a protein and a lipid sort of all made together. Okay, so the Golgi apparatus, at a smaller level, processes things, tags things, receives, sort of ships and sends proteins where they're supposed to go. Reads the tags, what type of molecule they are, packages them, and ships them out. So I like to think of the Golgi as like the UPS. Okay? Yeah? That's what you said? Okay, good. We're on the same page, Smo. So packaging and shipping division, or UPS, of our little manufacturing plant of the cell. Okay, so here's the rough ER. Those proteins that are newly made get packaged in vesicles going through the ER. The vesicles get pinched off and they come to the Golgi. Now notice these are fat, these are phospholipids. So a vesicle going up with another vesicle is going to merge. The fat phospholipids are going to merge together. Takes in those proteins. The Golgi has enzymes and machinery that can sort those different proteins where they need to go, repackage them into new vesicles, and send them on their way. So literally it's kind of like the UPS of the cell. And you think, wow, that's magic. How does it know? There's a lot of enzymes, there's a lot of proteins and machinery. And what we don't talk about a lot, there are all throughout the cell, there are tracks. Your cell has like train tracks. And in the beginning of this, I showed you pictures of fluorescence and confocal microscopy, where we tag proteins with fluorescent tags. And some of those cells you could see there was like strips of stuff going all through the cell, right? Like green and blue. Those are microtubules. These are the cytoskeletal proteins that make up almost like the skeleton inside of the cell. And there's motor proteins. It's crazy because they have little feet. I'll show you guys a video of them sometimes. They're little feet, and they attach onto the little vesicles, they grab on, and they walk along the microtubules. I'll show you a video later, they're cool. They're like, oh, actually, did I show you guys the inner life of the cell or not yet? That video? Okay, I'll show you that too. But they actually walk, and they're the ones that pick up, grab, and transport it. They walk hand over hand, and they transport the vesicles all throughout the cell. So it goes from ER, it goes from Golgi, it goes out to where it needs to go. Now, a lot of these proteins will be destined to leave the cell. They're making either receptors or proteins or molecules that need to exit, hormones that need to actually leave the cell. These will be transported up the membrane. They'll undergo exocytosis of the membrane, and then those newly made proteins or glycoproteins will actually exit the cell. So microtubules with motor proteins allow the magic of sort of moving and getting these sorted and shipped where they need to go. I will show you in the video the spot where we can see that. It's very, very cool. And for me as an undergrad, when I was learning about all this, I would always think, how does that organelle know how to do that? Or how does that cell know? It's like magic. How does it? And a lot of times we can explain it with the enzymes, ATP, and motor proteins walking along microtubules, because they will take everything sort of where they need to go. All right, mitochondria. I would say I hate the PG&E. I hate PG&E. Terrible, big monopoly, horrible, expensive. I hate it. Every time I have to make a bill to PG&E, I'm just like angry. But the mitochondria is like the PG&E. Okay, it's like the powerhouse of the cell. It's generating the electrical energy of the cell. Most of the cell's ATP is coming from all of its mitochondria. And it's the most complex of all these organelles that we're talking about, is the most complex. Really, it kind of came about early in history. There was a bacteria type cell, and it was a prokaryote, an early bacteria, aerobic bacteria. That means it used oxygen to make a lot of ATP. And an early eukaryotic cell, which is basically the lineage that makes up all of us and plants and animals and fungi and a lot of other things, engulfed that. This isn't to scale, but this prokaryote is very, very small. Engulfed it and actually didn't consume it. It stayed alive and gave it a protected house because of its selectively permeable membrane, gave it a space of safety, and the early prokaryote was able to, as long as oxygen was provided to it, churn out and make a lot of ATP. So it became sort of a symbiotic relationship, an endosymbiotic relationship where one takes in something else inside of it, and now they kind of co-evolve and live together happily ever after, because that early bacteria became the mitochondria that are in multicellular organisms. And actually now we can trace it, and they're right here. Here they are right here. They have the same DNA as a lot of bacteria, prokaryotes. They have circular DNA. They have their own DNA. They don't have the DNA that's in the nucleus of the cell. They actually, mitochondria have their own circular plasmid DNA. They replicate on their own, like little amoeba. They just pitch, and you get two, and you get four. So separate from the normal cell cycle that we're going to talk about in a minute, they do that on their own. So they're really abundant. You see cells that have a lot of mitochondria in cells that need a lot of energy, like muscle cells and sperm. We'll see lots of mitochondria because those cells need a lot of power output. They burn, they need a lot of energy. Interestingly enough, the egg, the human egg, is loaded with mitochondria and protein and ribosomes and enzymes, a lot of mitochondria. And it's ready for division of the embryo if it gets fertilized. All of the mitochondria that you inherit, that you have in your entire body, came from your mama. Okay? Mitochondria is inherited through the maternal side in the mother's egg. I can trace your ancestry, your lineage, all through the maternal side, even back to like the Eves, early like Homo sapien Eves, through mitochondria. Because the genetics are, those genes, those mitochondrial genes are passed down and give us lineage and ancestry tracing. That's what a lot of these companies do actually. They can even look at some of the DNA, including your mitochondrial DNA. So you get all of your mitochondria and mitochondrial DNA from your mom. So it's pretty interesting. Genetically speaking, it's a really big deal. So we called mitochondria the paraplant, or the PG&E. Simple structure, okay, so not so simple, it's a little more complex than the others. It's got a double membrane structure, so it's got the outer membrane, and then it's got this inner membrane that's highly folded. And then it's got the matrix, the gelatinous matrix of the inside. Now inside here, it's got studded all along this inner membrane. Tons of enzymes. Remember that ATP synthase that I made you guys learn about that makes all the ATP? All those enzymes, the electron transport chain with all those complexes, the enzymes, the machinery, the ATP synth, they're all studied inside of there. Okay, so it allows the mitochondria to keep carrying out that aerobic respiration as long as it's alive. There's a picture of an actual one. This is through SEM, cryofracture. They split the cell, they froze the cell, cracked it in half, and then pseudocolors we can actually see. Here's the inner membrane, or cristae, and then the outer membrane. So all those enzymes of the aerobic respiration that we talked about are all right within there. And then if we were to see it, it's not real present right here, but their plasmid DNA, it's circular DNA. Basically they have circles of DNA, just like bacteria have. We have DNA that's linear and parceled into chromosomes. Bacteria, prokaryotes have circular DNA just like our mitochondria. Okay, so it's neat. We can use it for lineage tracking. Lysosomes and peroxisomes. These are vesicles, membranes, sacs or vesicles that have specific functions in the cell. To break down toxic waste like alcohol and free radicals, and lysosomes break down food type stuff. They're loaded with digestive enzymes. So they have a lot of involvement in digestion and breakdown of poisons. Think of poisons, peroxisomes. Lysosomes, they have digestive enzymes, many times dozens of enzymes loaded in the lysosomes that allow them to break down all those different macromolecules that your cells come across that you consume. Proteins, fats, polysaccharides, nucleic acids. Lysosomes have the specific enzymes, lipases, proteases, carboxypeptidases that break down all those macromolecules, okay? Tay-Sachs disease is an example where the individuals are missing just one enzyme that breaks down fats. And so in their lysosomes, they're missing this one critical enzyme, and because of that, they end up building up a certain amount of lipids in the brain, and it can be most individuals, if they're born with Tay-Sachs, they die very early. So brain degeneration, a loss in sort of developmental progress and cognition, all that, because they get fatty lipid accumulations in the cells of the neurons of the brain. I don't know if you read the opener to the chapter, but it talks about a couple and their child has Tay-Sachs. They don't typically live longer than 5-6 years old because it's that severe. They're lacking just a key digestive enzyme. And because they don't have it, those macromolecules build up and they gunk up the cell type, in this case neurons, that are needed for brain function. Lysosomes also use enzymes to recycle organelles and macromolecules. So your cells are recycling organelles and parts. They're breaking them down and using the pieces so they're not wasteful. Okay? So all those are done in what's called autophagy. Autophagy, phagy or phagocysis is to eat. Auto is what? Self. Eating yourself. Okay? So they will digest and break down the organelles and the deteriorating macromolecules and big structures and break them down and use the parts. So they're used for digestion of non-self and self. Macromolecules and falling apart organelles. Peroxisomes are really more the toxic waste removal system or the poison decontaminators. P for poison, peroxisomes. They're membrane-walled vesicles or sacs of oxidase enzymes. Okay? Oxidase enzymes help to break down and neutralize free radicals. Remember we learned about free radicals, right? Yeah. Unpaired electrons, they're the exhaust from aerobic respiration. They accumulate with mitochondrial cellular respiration and exposure to exogenous things like UV irradiation, for example. So the enzymes in the peroxisomes help to cut down and get rid of those free radicals. Okay, so they're good. They also break down poisons like alcohol, hydrogen peroxide, those things that actually are also either taken in or products of metabolic oxygen pathways. Okay? And they break down long chains of fatty acids as well. So I would say the peroxisomes are like the toxic waste removal system. They're really numerous in the liver and the kidneys. Your kidneys are filtering all the blood plasma, all your fluid, every minute of a daily cycle. And your liver is filtering and detoxifying all the wastes and the poisons and just filtering all of your blood as well. So those two have the highest amount of peroxisomes inside of themselves. So if we look at lysosomes, digestion, and autophagy, we can see here's the little lysosomes, little vesicles. They got all the digestive enzymes in them. You take in, by phagocytosis, you take in a bolus of food, or your cells are sort of taking in by phagocytosis a bunch of macromolecules in a food vacuole, so by phagocytosis. The lysosomes will fuse with them. Again, they all have phospholipid membrane, they can all fuse together, and break apart those larger macromolecules into smaller monomers that can then be used for fuel and broken down in the pathways of metabolism inside the cell. This is a mitochondria is getting rusty, it's falling apart, it's old. Peroxisome, maybe it's done its job, it's kind of fallen apart. The lysosome will fuse with those structures in a vesicle and allow the breakdown of its self-components to get rid of them and recycle the parts. And this happens a lot with things that are degenerating and falling apart. Autophagy will actually be a part of that process. The last sort of component here of organelles that I want you to know are the centrioles, or this whole thing is called the centrosome, but these are the centrioles. And if we look at them, they're just right there inside the cell. They're usually off at kind of one end of the cell. They're made of microtubules. So the cytoskeletal, remember I said they're like railroad tracks, those little cytoskeletal proteins all throughout the cell. These are the same structure, except they're made of 27. They're like little star arrays, and they're short. And if we look at them in cross-section under the real microscope, you can see they're little sets of 3, 3, 3, 3, 3 in these cute little structures. To me, they look like churros. So I just say these are the churros of the cell, but do not let that undermine their very important role. The centrioles actually allow the growth of microtubules. They will polymerize. Microtubules will grow off of here, growing off in all different directions. They're the ones that actually grow the mitotic spindle, this big spindle that's made of microtubules. And as we're going to see in a minute, that's what allows the cell to divide its chromosomes and split in two. So if I take out the centrioles, our little cute little churros, I would not be able to have that cell make a mitotic spindle. It wouldn't be able to undergo cell division or mitosis. And it's also critical for making cilia and flagella. So sperm that grow a tail or a flagellum, it comes from these little centrioles. They grow the microtubules right out from them. Same thing with cilia that we looked at in like the trachea. So really, really important, this little gadget, this little organelle, this structure. Okay, I'm gonna not have us do this right now. I'll have you do this as homework and kind of work through it, and then we'll talk about it in class. I don't know if have you already done this? Did you look ahead? No? Okay. One was very dehydrated and unconscious, and an inexperienced friend, little knowledge of the human body, desperately was trying to help her. She gave her an IV containing only water. Hey, I surveyed my own friends just to check their biology knowledge here. She gave her an IV only water. Was this a good decision? What do you think will happen to the woman? We could just say it right now. You don't have to do this as homework. This is kind of related to the other material, so I was hoping you looked through and read this. I had a similar question on the exam. Sort of. Do you remember?
So vitamin D deficiency.
What?
Is that what you're talking about on the exam, vitamin D deficiency?
Nope.
Is it like what her blood cells will lyse?
Okay, so this is exactly, when you're getting into the lyse, that's where I want you to go. We talked about tonicity, how concentrated solutions are and what that will do to cells. Specifically, that refers to osmosis, the movement of water, right? Osmosis, okay? If you put cells into a solution that has not a lot of stuff, matter of fact, it has no stuff, but my cells have a lot of stuff in them. I'm putting them in a solution that's called what? This was on the exam. I'm putting them in a solution that has very little stuff, so we would call that isotonic, hypotonic, or hypertonic? Hypotonic, okay? Hypo. Less stuff, hypotonic. Good, dude, you're like, I got it, okay. And inside the cell, it's hypertonic because there's more stuff, right? Okay, so now in that scenario of no stuff, now bathing the plasma of my red blood cells, where's the water gonna go?
Inside the cell.
Yes. It's gonna go from high water outside to low water inside. Come into the cells, it's called cell swelling and lysis. They are going to lyse. You kill your friend by doing this, potentially. Yes? Okay. People think, oh no, water's good, you drink water. Yeah, no. You drink water through your GI tract, and then you absorb the macromolecules and the H2O that you need, and it gets all filtered by your body. It goes straight into your veins and your arteries, and you're flooding those cells with the hypotonic solution. This is why IVs, it was brilliant to figure this out. What's the tonicity or the solid concentration inside of blood? Okay, well, when someone loses a lot of fluid, a lot of blood, we have to match that exactly. Even if we're just giving them fluid, no cells, just fluid. And we have to have salt and sugar in there in the right proportion to match the actual blood so that your cells don't shrivel and they don't lyse. Yes? Okay, so I had a question, I had a little picture of that on the exam, like putting it into different scenarios, what would happen. This is basically an application of that. Okay, cell division mitosis. We'll go through this just a few minutes. Some of you have already had exposure to mitosis. I really don't want to spend a lot of time on it. Just to recap though, it's really important. It's good for reproduction, right, and development, growth and development of the embryo, the fetus, you throughout your entire life. Your cells are all undergoing mitosis and regeneration and repair. So when they slough off and they die and they get damaged, you have cells and tissues that are replenishing so that you can continue your life and survive. So reproduction and making cells that support sperm and eggs and allow them to grow in the right way, grow and develop through embryo and fetal development, and rejuvenate things like blood cells, white blood cells, red blood cells, a lot of the stuff we did in lab last week looking at. We have chromosomes that are passed on. All of your cells have DNA that's contained in chromosomes, right? This is a little bit of review. We have identical sister chromatids. That means this is when they're replicated. So when DNA replicates, really a chromosome is like a long, thick, packed, condensed set of DNA. When it replicates, like before it's going to divide, I go from this one set right here, which is a complete chromosome. Don't let the X confuse you. We always say chromosomes are like this. Really, I have everything I need on this one chromosome right here, okay? And then when I replicate that DNA, there's enzymes that copy that. Now I have an exact duplication. They're tethered kind of at the waist, which we call the centromere. So when I replicate my DNA, they're tethered right there. That's a duplicate. We call those sister chromatids. That's easy to remember. They're sisters. They're little identical twin sisters, just sister chromatids. You have to replicate your DNA or your genome before a cell is going to divide and split into two daughters. I love all this is it's daughters and sisters, okay? Before it's going to split into two daughter cells, you have to replicate the DNA. Otherwise, what would happen? If I just had my one chromosome and I didn't replicate it, but like I divided into two cells. Yeah, I would half the amount of my DNA. Like that cell would now only have half of the genetic information. It would not be able to function, right? That happens, but it happens in the gametes, the sperm and the eggs. So it's a very specialized process. We're going to talk about that later. That's meiosis, if you remember learning that. So this, it has to replicate the condensed chromatin. The DNA coils around the proteins, condenses into the chromatin structure, and it's distributed to the new two daughter cells in division. So these will actually pull apart right at the centromeres. So you have a whole chromosome here, or sister chromatid. Now it's on its own, so it's a chromosome. And then a whole chromosome over in the other cell. Okay, so the cell cycle, and here's how I remember. I remember the cell cycle. Interphase is all the stuff in between when the cell's not divided. Interphase is here. There's G1, S, G2. Growth. S stands for synthesis. This is when the DNA is replicating, when the chromosomes are replicating. G2 is final preparations for dividing. A little bit more growth, housekeeping, just getting everything ready. And then M phase is all this right here. So it's interphase, M phase. These are respectively broken down into G1, S, and G2. But the business end of when a cell is actually dividing is proportionally a small amount of its life. Usually it's in interphase, doing its thing, metabolizing, breaking down, doing autophagy, doing phagocytosis, digestive enzymes, just doing all of its machinery. And then when it gets the right cues or triggers, or it needs to replenish tissues that are damaged, it'll get triggered to enter mitosis. And I remember the stages of mitosis with this mnemonic. I pee on the mat. That's gross. I know. Just think of like a dog or something. I pee on the mat. But on the sideline, okay, it's irrelevant here, but interphase, pee on the mat. Cytokinesis is the very last step we'll look at. C doesn't work in my little acronym. Mitosis phases are prophase, metaphase, anaphase, and telophase. The last one is cytokinesis. What does cyto stand for? Cyto. Cyto means cell, okay? Kinesis is like development or creating, right? So we're developing, we're cutting now that cell, that cytoplasm of that cell into two cells. So its cytoplasm is dividing right at the end. So mitosis specifically refers to what the chromosomes are doing. Cytokinesis is referring to what the cytoplasm or the cell itself is doing, and actually is pinching into final two daughter cells. All right, so remember IP on the map, and you'll remember that. Oops, there's the I actually wrote that. Okay, I know it's weird. You guys can tell people what I tell you. Why is she telling me those things? I remember these things that way though, that's why. All right, mitosis, what it looks like is, here's interphase. It's got the nuclear envelope, it's got your centrioles. Centrioles will actually duplicate themselves. They'll generate more microtubules. Remember the little churros? The churros will replicate themselves, so now you got two pairs, and they're almost always like that. They're like in that perpendicular little arrangement. Now you got two, so they're like, hey, we're getting ready for mitosis. We gotta break down the nuclear envelope, we gotta duplicate the churros, we gotta set up the mitotic spindle. The mitotic spindle being this really important structure of microtubules that branch, grow out from the churros, from the centrioles. Those would allow the microtubules to grab on to the outer side of the duplicated chromosomes. Once they do, and it's hard to see, but they line up, they grab right on the outer side of those chromosomes, okay? So that with the right enzymes, and again, they're motor proteins, they walk on the microtubules and they cleave ATP. So they actually like walk. They play a little tug of war where they walk along the microtubules and they rip the chromosomes apart. So the chromosomes get ripped apart, the sister chromatids. So you've got one set of sister chromatids or chromosomes going to that pole, this one going to that pole. When we actually put these into phases, when the chromosomes are distinct, they're condensed, they're replicated, and we can actually make them out. We can see them under the microscope like this, and we'll look at that in lab on Thursday. We didn't have time last week, so I'll have this set up. You can look at these cells. We can actually see them. That's prophase. When they line up on the metaphase plate, this is called the metaphase plate or the mitotic equator. They line up right in the middle metaphase. That's metaphase. They line up right in the middle. They get ripped apart. They go to the two different respective poles of the cell. That's anaphase. Now that there are two ends, you actually have a cleavage furrow where the cell starts to pinch in like you're putting a cord around a balloon and you're tightening it. The muscle fibers inside of the cell, they actinize and start to pinch, pinch, pinch. And the two different sets of cytoplasm go to this daughter cell and this daughter cell, culminating finally in two official cells that start out exactly sort of how you started out in the beginning. You didn't lose any material because you replicated the chromosomes before you actually underwent mitosis. And actually, some of our slides, they look just like this. So this is from a light microscope stained. You can see little chromosomes all in a line right here. You can see them kind of ripping apart right here. You see that? Those are metaphase and anaphase stages of an embryo undergoing these different phases of mitosis. Okay, here's another visual of what they look like.